Welcome back, everybody. Today I wanted to take some time to talk about whole genome sequencing for bacteria. So when I was a PhD student, I took part in this workshop where we had brought a bacteria in, monoculture, and then we sequenced the whole genome of it. It was just kind of a practice for us students to get another side on how the lab part of the analyses work. And it was really good. And after that, we ended up finding out that it was a novel bacteria. So we have here a novel species, and yeah. So if you guys find these videos helpful, I know I don't like doing these plugs, but if you find them helpful, go ahead and like the video and then subscribe, and I'll have an idea of what kind of videos are more helpful. If you guys want to see more tool explanation videos, let me know. I really like the more analysis side, so just using the tools. But yeah, feel free to let me know what you guys like. So jumping into this, we gave it a new name, so Methylorubrum endophytica, and we found out after all the analyses and stuff were done that it was actually novel, like I'd mentioned before. We isolated it from soybean seeds. The whole project was from surface sterilizing the seed of soybean or other tissues, and then just extracting the bacteria out of the inside of the seed or tissue. So those are endophytes living within the plant. To download this, you can do the exact same thing like we did before. If we click on this SRA run down here at the bottom, we can see we have 150 base pairs for the forward read, 150 base pairs for the reverse read, GC content of 67.8, 68, as well as a quality graph here. And we can see that there's a lot of really high quality reads in here, which is going to be helpful later on when we build a new genome, because we want to set our threshold at 25, or even sometimes people do higher than that. That way you're really confident in the base pair calls that you're using in order to build the genome, since you don't have a reference to map against. Like I mentioned, we have paired end. So that's something that we'll have to pay attention to when we trim, as well as feeding into Unicycler, which is going to be the assembler that we're using, the de-novo assembler. All right, so to download, just like we did before, you can do CD. I already made the folder, and it's called Bacterial Genome. We go in here, raw reads, and then I already extracted it too. So we can remove star.fastq. There we go. So when you download this, you can do, what I did is prefetch and then srr 93211164. And what you'll get by running this is this folder. So this first darker green is going to be a folder, and inside that is this srr.sra. I just moved it to the current working folder of raw reads. That way when I extract it, everything comes into the raw reads folder, and then I'll have to move more files later on. To extract from the Sra file, we can do the same thing that we did with the RNA sequencing files. We can do fastq dump, and then we need to split the files. So we know that when we downloaded them that we have paired end reads, so we need to extract them accordingly. So split files, and then we can feed in our SRA, and what it's going to do is it's automatically going to find the forward read, and it's going to find the reverse read and put them into the two separate files, like you can see above here. And we'll wait for that to finish, and there it's done. We do ls now. Now we can see we have, again, our accession number with our forward reads and our reverse reads. So if we move back up, what we can do is run fastqc. So right now I'm in base. I'm not actually running in an environment, and later on that'll come into play when we start doing Unicycler, because Unicycler, the one that installs by default, does not work. So I reverted to the one that I used before I submitted this to NCBI, and it did work. So I don't know if there's something not happy with the most recent version of Unicycler. So it's a good thing that we were able to go back and install those. So let's run fastqc, and we'll put the results in, we'll put them in the same folder, raw reads, and then we'll feed in our read files, raw reads, and then srr whatever star.fastq. So what this is going to do, it's going to look in raw reads, srr, it's going to find that accession, so anything that starts with that, and then it has to end with fastq now. So it's only going to be our read files. We can also do -t, and then we can do 2 threads. So it'll just run both of them in QC at the exact same time. This runs a lot faster than the RNA sequencing ones did, and that's just because these files are much smaller compared to the RNA sequencing files. And just like that, it looks like it's done. We can go into our raw reads folder, and then let's do multi-QC. And then the dot just means in this working folder, which is raw reads, and then it should automatically find, yep, our 2 reports. Let's go ahead, I'm gonna open them, raw reads. I'm gonna open multi-QC as well as an individual file. So you can see here, the number of duplicates is kind of high. GC content, so that matches fairly close with what the NCBI page said. The NCBI page said 67.8, I believe. Yep. And here we have 67, and then 2.1 million reads, which is good. We can see here that the reverse read actually has a higher quality throughout almost the whole sequence than does the forward read. And like I mentioned before, we're going to have to chop this at a little bit higher because we want to be more sure that our assembly is correct. So this is why I took this guy, and so we can see here, I've ran it 2 different times, and that is what I'll show at the very end up here. One time I did a head crop, so I chopped off the first 15, which is this noise that we see. And then another time I left that in, and I only did adapter removal, which it doesn't look like there's any adapter content at all, but just to be sure. But otherwise, it looks fairly good. When we remove reads, we're going to set it at 25, so we'll get some really short ones in here, but like I mentioned, we'll be more confident that the final assembly is a better quality than keeping all of the reads, which could introduce some more error in the assembly anyway. So you can see that's the final result. So actually, let's now trim. So we can CD and we'll back out, go back to our home folder, Bacterial Genome. So we'll trim now. So just like we actually need to bring in, we do ls, we just have our raw reads folder. So we have to do 2 things. We have to make a directory for our trimmed reads. Okay, now we actually have to also copy in our adapters. So if we go back to here, and then back, here we go. So Nextera. So let's go back here. We'll copy in our adapters and we'll select which one we want. So I'm in root. I installed Conda in root, so when I installed Trim-O-Matic, that is also in root. So we have to do root, and then mini Conda 3, and then share, Trim-O-Matic, and then adapters. So if I press tab a couple times in here, we get all of our adapter sequences, and I'm gonna use the Nextera. So even though it didn't look like in the QC file there were any adapters, I'm still gonna do it just to be safe. And then Nextera, and then just like before, if we do a dot, it's just gonna say copy this file right here. If we do ls now, whoops, we have our Nextera adapters, raw reads, and our trimmed reads folder. Awesome. We can do Trim-O-Matic. We want paired end because we have paired end reads. Threads, it doesn't really matter. We'll do 8. Trim-O-Matic is really fast since this is a really small file. We'll feed in our reads files. So reads, sr, we'll go _1 for forward. What is it not liking here? It's because I put all the QC in the same folder instead of moving it to a different one. So then we'll do raw reads /srr _2.fastq. Okay, so we're feeding in our reads. Now we can set a base out. What that's going to do is it's just going to tell Trim-O-Matic where we're going to put our reads. And then our base out will feed them into trimmed reads. And then we'll give them the same name as we have here. So 9321164, and then we do .fastq. What it's going to do is it's going to parse out this .fastq, and it's going to put, just like with the RNA sequencing trims, _1, _2, or and then P or U, depending on if it's paired end kept, or if it had to remove the other reverse or forward read for that particular read. All right, so now Illumina clip, we're going to remove our adapters, Nextera, and then 2, 30, 10, and then instead of 2, like we did with the RNA sequencing, going to do 8. So that's a match length of 8, I believe. And then we'll keep both reads. Unlike the RNA sequencing, I here am going to do head crop. And then we're going to chop off the first 15. So if we look back at the QC here, this is really not good. So I'm just going to chop it off at 15 and say, okay, starting at base 15 or base 16, if we do a sliding window afterwards, hopefully our quality is better. So if we go back, so that's head crop, and like I just mentioned, we'll do sliding window. And for that, what we're going to do is we're going to do a window of 4 base pairs, and we're going to look for a quality of 25. If we go back up here, we'll do 4 base pairs at a time. And as soon as those 4 base pairs, so 1 through 4, 2 through 5, 3, 6, so on and so forth, as soon as that average of those 4 base pairs drops below 25, we're going to cut the sequence, and then we're just going to throw the rest of it away. So since our sequences are 150 base pairs long, 151, we can kind of afford that a little bit more than if we only had 50 base pairs. So with the RNA sequencing, we only had 50 base pair length. And so by doing the sliding window, we could really run into some problems. The same with the head crop. It's good to do, you just might throw away a lot more sequences. If we go ahead and run this, it should be pretty quick since our files are really small. Awesome, so that's already done. We can see that forward and reverse surviving is about 94%. Only forward is 3, and then we dropped 0.25% of reads, which is good. Now comes the interesting part in order to get Unicycler to work, or that I found in order to get Unicycler to work. We have to create an environment, so we can do conda create, and then we'll give it a name, and we'll call it the same thing, so we'll call it Bacterial Genome. And then we actually have to set it to Python 3.5. So if we do conda search Unicycler, we'll get a list of all of the versions of Unicycler that are available through conda. And the one that I used before I submitted it to NCBI was 041. And so here we're going to install the same one. And then to install that one, we have to create an environment that is Python 3.5. In order to create an environment of a particular version of Python, we can do Python equals 3.5. If we run that, I already set it up, but we'll do it again. When we run that, now if we try to install Unicycler, it's going to look for the most recent one for that version of Python. And when this is done, we can scroll back up. And I think it's actually one newer than the one that we are going to install. So we're going to have to specify 041, otherwise it will install 044. All right, so now our conda environment is done. We can head on into it. So conda activate, bacterial, let's see, no, it doesn't autofill. Bacterial genome. All right, so now on the left-hand side, we can see bacterial genome is the environment that we're in. And then if we scroll back up, we want to install Unicycler version 041. That's the one that I had used on Galaxy. Galaxy is super handy, but conda install, and then Unicycler. We need to specify which version. So we want 0.4.1. Go ahead and start installing. We'll say yes. Some other things that I noticed when I tried to get Unicycler to run even this way is that it got really angry, and this might just be my computer for whatever reason, with this pylon version 1.23-2. So I had to install conda install. We'll do conda search pylon, and we'll look at all the versions that are available. I had to install 1.22, which is this guy here. So conda install pylon 1.22. We'll get that going. In addition to pylon 1.22, had to install Java JDK. So conda install Java-JDK. And then after this, I was able to get everything working. All right, we're inside our environment. We have our JDK installed, we have our pylon installed, and we have Unicycler 0.4.1. Now, let's begin assembling our genome from our reads. We do Unicycler -1. Well, let's look at the menu over here. All right. So how Unicycler works is it basically finds a sequence, and if there's a branch, it'll start moving after that to have diverging regions. And a reason why long read sequencing is really helpful for de novo assembly is, let's see if they have a picture, is here. You can have your reads map to your long read sequences, and if there's a gap in there, then you know more of how long your genome should be, and then your short read sequences are going to be better quality, so you know with your final contigs, which are shown with the green, your final contigs will have a lot higher quality. So here you can see that there's some gaps in here. Your contigs will have higher quality, but your long reads will give you that template of what to go off of. So with Unicycler, we can feed in our forward read from Trimmomatic, we can feed in our reverse read from Trimmomatic, and additionally, we can feed in our unpaired reads. So if we go into our Trimmomatic folder, let's actually back up, CD, and then we want to go to trimmed reads. In here we have our forward unpaired and reverse unpaired. We can merge those together since they're not matching. The reads in 1U don't match up with the reads in 2U like they do with the paired end reads. If we do cat and then srr*u.fastq, that's going to find 1U as well as 2U, and then we can feed that into srr, and then we can just make a new file called unpaired.fastq. Now ls feeding into Unicycler -1 is going to be this one, our 1p. Our -2 is going to be 2p. And then our -s, or our single or unpaired reads, are going to be this unpaired file that is from our 2U and our 1U. All right, so let's start building this out. So Unicycler -1, we want our trimmed reads, srr1p dot fastq-2. We'll do srr, nope, trimmed reads, srr underscore 2p dot fastq. And then we can do dash s, srr unpaired. Now we're going to use all of the trimmed reads instead of just the paired end, like we could with star. We don't have long reads that we could build a template out of. Let's point to out, so dash O, we'll just have it make a new folder called assembly. Verbosity, I like to set it at 2 because then you can see everything that's going on in all the steps and kind of judge better of how much it has left or how much it has done. So we'll set it to 2. It can go 0 all the way to 3. 1 is just the basic, 2 is a little bit extra info. The min FASTA length that I used when I ran it before with Galaxy was 150 base pairs. However, when you submit to NCBI, you have to delete everything less than 200. So let's just go with min FASTA length. There we go, LENGTH, and we'll set it to 200. That way, whatever comes out can just be, that assembly file can just be uploaded right to NCBI. Additionally, we will do -t and 64. The first part of the assembly with Unicycler doesn't use all 64 threads. It'll subset that down to 32 and then run with 32. But the later on, when it starts doing the error correction, it has to remap all of the reads to the genome, or to the quote-unquote genome, the contigs or contiguous sequences. And by doing that, it will use all 64 reads. Mode, we'll just do normal. So that's default. Yep, you can see here, default is normal. So we actually don't have to set anything. And then linear seeks, we'll just keep that at 0 as well. If you really want to dive into some optimizing, what you can do is come down here with the advanced options, and these are more fed into Spade and then the other tools too. So there's Pylon, which we had to install. Spades is the main one. And for short reads, basically what Unicycler is just a Spades optimizer. So that's mainly what we're gonna do. We don't have long reads, but yeah. So we should be able to just let this go. Let's see if it starts out. See if it gives us an error. There we go, it actually started. So I'm gonna let this run, and I'll fast forward through it so you guys don't have to sit and wait. All right, now we have a completed genome assembled. So this is going to be a contig, or as complete as we can get. It's not going to be a whole chromosome. If we scroll back up here, we can see that we have a total of 2 components. So this bottom one is probably going to be, since it is complete, it's probably going to be a plasmid within that bacterial sample. Whereas this top one here is going to be most likely the chromosome of the bacteria. And then we have the total length up here. The number of segments, that's going to be all inclusive, I believe, with all of these, not actually removing the short ones until the end. If we look at the assembly file, here we have we scroll all the way down to the bottom, we should see how many contigs are in total. So there are 118 contigs. All right. The next thing that we can do is Prokka, which is going to be the Prokaryotic annotation. So we have to exit out of here. That's not what I want. As we can CD back into bacterial genome, but we can do conda deactivate. Because Quast, for some reason, will not install for me in this bacterial genome. It just hangs and it'll continue waiting over and over and over and over. I have Prokka installed, which can be installed by just doing conda install Prokka, and then Quast too. So Quast is going to be the quality assessment tool for the genome. In order to run Prokka, so you can do Prokka-H. We have a bunch of these options in here, but the main one that we're going to do is just the output directory is the only option. There's a lot of other things in here, but for a simple annotation and CPUs, but for a simple annotation, it doesn't really matter too much. Can dive into it further down the line too. So if we do Prokka output directory, we might it should make one output directory, and then let's make sure that's right. General outdir, yep. We'll call it Prokka results. And then we can feed in some of the other kind of stuff. So we can give it genus and species if we really need. We can do genus and then methylorubrum. And do strain. Since we don't have a species, I can just call it Q1. Kingdom is already bacteria, so that really doesn't matter. I don't think these change anything, the genome and the strain. It's just information in there. And then finally, we can do CPUs 64. This one will take a little bit to 2. It's not as snappy as that Trimmatic was. Assembly, and then you can just feed it in our assembly FASTA file. So this is going to go through Prokka. It's going to find all of the possible genes that are in this contig assembly. It's going to put it in our folder Prokka results. It's going to have a bunch of different gene formats in there. So we'll have a GFF file, we'll have a table, we'll have a bunch of good stuff. I think that's all what's down here. And then strain, CPUs, we'll give it everything, and we'll just let her go. It takes a little bit of time, so I'll speed through this as well. Great, now we have Prokka done. If we look at some of the files, or a file from Prokka, let's see, let's choose this one. This might be the whole genome. Yep. So let's FNA, let's F amino acid. Here we go. So now we have all of our genes, predicted genes, hypothetical proteins, in here. So we can look for something like flagella. Here we go. So we have a flagellar protein there, flagellar. We look for 16S sequence, 16S, pre-16S, 16S, pre. So it looks like these are proteins. So 16S rRNA isn't going to be in here, but alcohol dehydrogenase. We have all of our genes now from the assembly that we have, which is good. The next thing that we can do is check the quality of our assembly. And to do that is with Quast. So we can run Quast help and see what all we have here. A lot of options, but Quast stands for the Quality Assessment Tool for Genome Assemblies, like I had mentioned before. And so the options that we'll use here are going to be -o, so we'll feed it the output directory. We'll just do something like Quast results since we did Prokka results. In G, we can feed in our features that we got from Prokka. So this will be our GFF file. If we go into cd, well, let's ls Prokka results and see what we get. So we'll feed it this, our GFF file. What else? Threads, we'll set our maximum threads. I don't think it'll take too awful long, but then we can also feed in our reads. Here we are. We can feed in 1, just like we did with the Unicycler. We can feed in 1, we can feed in 2 for our reverse reads. We can't feed in unpaired though, I don't believe. We can try it. We'll try feeding in single end. And then we can also set our gene threshold, which will, let's see if we can find it. It's dash one of these options. There's gene finding. We'll set gene threshold. Not sure where that's at in here. That was an option that was set in, here we go, comma separated list of threshold lengths of genes to search within the gene finding module. We'll do 0 and 100. That's the default of what Galaxy used. So we'll just repeat that here, and then we'll feed in our assembly too. Additionally, we'll feed in the flag for Glimmer. Let's see if we can find Glimmer. Here we go, which is going to use Glimmer HMM for gene prediction. So there's another option instead of Glimmer. I don't remember off the top of my head what it's called, but you have to have a license, I believe, was like the warning that it gave me. So we'll just use Glimmer. All right, so we'll do Quast, and then -o, we'll just do Quast results like we did with Prokka. Quast results, -g, this is we're gonna feed in the output from Prokka. We'll do Prokka results, and then Prokka.gff, so this is gonna be the annotation file. -t, we'll do 64 threads, and then we can feed in our reads. And we'll do -1, and we'll do trimmed reads SRR 1P, so a paired end. -2 is gonna be trimmed reads SRR 2P, so the other paired. And then let's try to feed in single, which is --single. And we'll feed in our merged unpaired reads, which is going to be trimmed reads unpaired. Okay, we'll set our gene threshold to what Galaxy used. Gene thresholds 0, 1000. So you just put a comma between them, and then we'll give it the assembly. So the actual config file that we have, and that's assembly.fasta. And additionally, from the assembly, we can do that Glimmer. What that's going to do is that's going to do some gene prediction stuff. So that we can go ahead and run that, and it'll fix the read names. So it expects /1, but when we get it from NCBI, it changes the read names to SRA numbers, so the sequence read archive number. Okay, now we have our Quast quality done. So we will come back over to here. So I'm going to open up the new quality. So this is one that I previously made using trimming, and we'll compare. It should be the same. So our reads mapped are going to be different because now we have our, we're unpaired in here too. So we have 5126, extended report, So in the spinal one, we don't see that. Predicted genes. But anyway, so this is going to be a map showing the length of our genome in comparison to the number of contigs that we have. So our first contig is going to be the largest, all the way down to our last contig being the smallest. So the first one has 440,000 sequences in it. So we had enough reads in order to get 440,000 continuous base pairs for a contig. Another thing that is kind of cool, if we go into here, we can see kind of the proportion of our contigs in here. So we can see our N50 is contig 12. If we keep going, they get smaller and smaller towards the end until we have a bunch of really small contigs here at the end. So of 46 contigs, we can make up almost the entire genome of our bacteria. I wonder why we don't have predicted genes in this guy. Or am I just not seeing it? Nope, I'm not seeing it down here. Anyway, so before when I ran it, this was the same thing. So this is using the same assembly pipeline, so trimming and all the way through, except here we have predicted genes. If you go to that Icarus thing, we should be able to, in here, I thought see genes, gene name. No results, so it must not work. But I also ran it again, but without using the trim, so without trimming the first part and then doing a sliding window too, and we have a little bit better scores here. So if we drag this off to the side, we can pull that over here. Look at this guy. Back. So if we compare these two, our longest contig is the same. However, if we look at the number of contigs, we have less contigs below our threshold. So here it's going to be 500 base pairs, or above our threshold. So 86 or greater than 500 here, where we didn't trim the first part, as well as didn't do a sliding window. And a percent mapped is also higher than trimming here. Our coverage, where is our coverage at? Average coverage is 59 where we trimmed, where over here we have 107 basically sequences for each base pair on average. We have a little bit more genes in the predicted genes, but overall it's not too much different. We have a little bit better quality here on the right, or not quality, longer, more full genome here on the right hand side than we do here on the left hand by less than 10 kb. So a couple contigs. So here the same contig 1 is the same size. So basically trimming the beginning, the head crop only affected the smaller contigs, which you can see. If we go to the Icarus, you can see on the right hand side here the length of the contig. So if you guys found this helpful, hit the like and subscribe, and we can see what we do in the future. Now that we have a couple different pipelines down with the RNA sequencing as well as the whole genome sequencing for bacteria, we can look at building a workflow using SnakeMaker, something like that. There's also Nextflow, where basically you put in your rules that you want. So initially you're going to have all of your reads be trimmed, and then you have to map your reads for RNA sequencing, or do Unicycler here to get an assembly and then do the downstream stuff. So that can all be handled in something that's called a workflow, and the workflow will let you basically move the workflow file to another folder and then run the same pipeline but on a different set of data. So it can automatically pull in the data from a folder. So as long as you have all of your files in the same raw reads folder, it'll be able to do everything else that you want it to. So yeah, thanks for watching.